Hi, good morning, everyone. Welcome and thank you for participating in this webinar, the Thermo Scientific webinar series. I'm Keat. I'm the Field Application Specialist in Apical-Scientific. So today we come to the last episode for our Thermo Scientific webinar series. But similar with the previous two series, we'll have a Q&A and also quiz after the slide sharing. Okay, and the gift for the quiz is very attractive, so do stay tuned with me throughout until the end of this webinar. Okay, yeah. So not to delay, let us start on today's webinar on cut and reconstructing of gene, CloneJet and RE restriction enzyme. Okay, so when I go through the slide, if you have any question, you can write in the chat box. I will try my best to answer you after the slide sharing. But if I can't answer you on spot, or if there is not enough time, maybe, so you can also email me at support@epicalscientific.com. Okay, yeah. Okay, so in today's webinar, basically I will go through the introduction on cloning, then what is the method and the workflow for cloning, and then I will share with you what is the enzyme that are needed upon the whole cloning workflow, and then also I'll share with you a little bit on the cloning kit and the expression kit and how it can help you in your study. Okay, and upon the whole sharing of the presentation, you'll find that I will share with you some tips and tricks on cloning throughout the whole presentation. Okay, so do take note on what I'm going to share with you. Okay, so let's start with a short introduction on cloning. What is cloning? Okay, cloning is a technique in molecular biology to insert a DNA fragment, which is your target gene or your gene of interest, into a vector, a bigger DNA. And then with this, it will generate a product we call recombinant plasmid. Okay, yeah. So the DNA fragment, which we call it as insert here, is usually the target gene which you want to determine is either from PCR amplification. Okay, so you determine the gene fragment maybe from PCR or maybe from digestion of from the genomic DNA. Okay, or from synthetic, generate from the synthetic DNA fragment. Okay, so means you request some manufacturer to synthesis the DNA for you. Okay, then you will stick, or the correct term, you will ligate the insert with a long DNA, which we call vector. Okay, there are two types of vector. The cloning vector, which usually is used to hold the insert, and mainly is for storage purpose. And another one is the expression vector, which mainly use is for gene expression in certain host cell. Okay, and the recombinant plasmid, which generate, is a double-stranded circular DNA. Okay, it's a double-stranded circular DNA. Yeah. So why do you need to perform cloning? Cloning, as everyone knows, is widely used across many different applications. For example, in molecular DNA, is to generate many copies of the gene, or to store a gene for your downstream application later. and it can also be used for gene expression study or study of a specific protein in a host cell or organism. Okay, in medical field, okay, cloning can be used to clone the cell and tissue to replace damaged body parts or use in genetic disease study. It can also use in agriculture, okay, to produce animal or crops with desirable traits, okay. In pharmaceutical, to produce a product for therapeutic protein or hormones or antibodies. Okay, so cloning is widely used in many different applications. Yeah, there are many methods for cloning. The commonly there are four. Okay, so it can be the restriction enzyme cloning, or we call it as sticky end cloning. Then there is TA or blunt end cloning. Also, there is gateway cloning and seamless cloning. But which method suitable for you, or which method you prefer most? Okay, so here's some tips for you. The restriction enzyme cloning or sticky end cloning, it will allow the gene to clone at the exact position, okay, or the direction that you want it to be in the vector. So this is very important for protein expression study, where you want to make sure the expressed protein is functionable. Okay, then the TA cloning or blunt end cloning is using the advantage where the Taq polymerase, they will produce a poly A tail in the PCR product. So you can directly clone the PCR product with a TA cloning method. But the drawback for this method is you will not know the direction of your gene in the vector. Okay, so it can be cloned in the opposite side. So it's either your vector will clone in the sense direction or in the anti-sense direction. Okay, and the gateway cloning, it is a site-specific recombination system which using the basic of phage attachment site, the ATTP, and the bacterial attachment site, the ATTB. So as a basic for the cloning. So you will need to make sure your PCR product flanking with the ATTP site, so that when you clone it into the ATTB site, then it will fix. Okay, so yeah, which means you need to design your PCR primer to carry this site. Yeah, okay. And for seamless cloning, it will allow you to assemble multiple fragments or genes into one single construct. So this is suitable to be used for multiple protein expression study. Yeah, so for today webinar, we will focus more on sticky end cloning and also blunt end cloning. Yeah, okay. So regardless the method that you're using for cloning, the basic workflow will be involve 6 steps. Okay, so you need to prepare your DNA fragment, which means to prepare your insert, and then there will be the second one. The second step is the DNA and modification, which you need to prepare your vector. Okay, then after you determine your insert and vector, you will need to analyze, quantify, or purify your DNA fragment. Then after that, only you can use them to perform the ligation. Okay, after ligation, you will determine the recombinant plasmid. So you will need to transform the plasmid into, because the plasmid amount will, after the ligation, will be very less. So you need to transform it into a host cell, okay, to get a multiple copy of this recombinant plasmid. Okay, and then only you analyze your clone, the colony that you determine. Okay, yeah. So I will share the detail for all these steps in the coming slide. Okay, so before you start your cloning study, you will need to decide on the vector to be used. Okay, but how to choose the vector that is suitable for your study? So here is some tips. First, to choose for the vector, you need to consider the insert size. Okay, some vector they can take up to 3 kb, some can take up to 10 kb, some that is possible to take up to 20 kb or more. So you need to know the insert size of your uh, gene of interest. Then only you decide what is the vector to be used. Okay, and then secondly is the copy number. Okay, copy number, it determines, it actually depends on the replication rate of the vector inside the host. Um, Usually a holding vector, they will have a higher copy number than the expression vector. Okay, so expression vector usually they will be bigger a little bit in the size, and they will have lower copy number. So it means bigger vector, it will have lower copy number. Is very common. Okay, so if your objective is to get multiple copies of the recombinant plasmid, then you go for smaller size or a simple vector. Okay, yeah. Next, you will also need to consider on the cloning side. Most commercial vector, they have the multiple cloning site where it is flanking by some common restriction enzyme. So you will find many different restriction enzyme in the multiple cloning site to ease the cloning process later on. Okay, yeah. Okay, next is the selectable marker, which usually is a antibiotic resistant gene that is carried by the vector. So this the presence of this gene in the vector is to ease the selection of the colony that carry the recombinant plasmid. Okay, so colonies that do not carry the plasmid, it may not able to survive in the medium that carry this antibiotic. So the most commonly used antibiotic, I believe, people who perform cloning before, you will know it will be ampicillin and kanamycin. Okay, then the next one that you need to consider is the reporter gene. So this is important for expression of the protein, so for expression study. Okay, um reporter gene, it will help to verify that a gene of interest is being transcribed and translated in a host organism or not. So if the reporter gene is expressed, it will suggest that the gene of interest is also active. Okay, yeah. And then lastly is the expression function of the vector. So if you are using a expression vector, most of the expression vector, they are specially construct to able to express a gene in specific organism. For example, like in certain bacteria or in human cell or in yeast cell, something like that. Yeah, so you will need to refer to the expression function of that vector to decide which vector to be used for your protein expression or for your gene expression study. Okay. Okay, so after you have choose your vector, then we'll go into the first step of the cloning, the DNA preparation step. So basically, this is to prepare the insert for cloning. Okay, yeah. So In this step, you need to generate DNA fragment or the insert, okay, that have compatible end that allowed you to ligate it to the vector that also have the compatible end also. Yeah, so the most common and effective way to prepare the DNA fragment is using a restriction enzyme, the RE. So later on when I mention RE, I will actually refer to restriction enzyme. Okay, yeah. So with this method, you can clone both DNA fragment in a specific place that you want it to be. Um, Restriction enzyme, or we call it as RE, okay, so it will recognize and cut at a specific DNA sequence, which usually is a 4 base or 4 to 6 bases long sequence, okay. So this sequence is called the RE site, the restriction enzyme site. Okay, so specific RE, you can see that specific RE, they will have specific RE site. Okay, for example, here, the Hindi tree here, the RE, this RE, it recognize the site with AAGCTT. Yeah, so when you read it from 5 prime to 3 prime is this sequence. Okay, yeah. So in another one, PST1, then you can see is CTG, CAG, something like this. Okay, so RE can actually generate a sticky end or blunt end at the DNA fragment, depending on the RE that you are using. So for example, here you can see the Hindi tree and PST1, they generate this sticky end. Okay, yeah, and then for Eco R1, it will generate blunt end. Okay, so yeah. And here are some tips to prepare your insert. So if your PCR product, right, you can design your primer to carry the RE site. Okay. Also, you may want to choose high fidelity DNA polymerase to avoid any possibility of SNPs or bias generate upon the PCR. Um, Anyone that attend the previous series of the webinar, you know what is the use of this high fidelity DNA polymerase. Okay, yeah, because common tag DNA polymerase, they will generate 1 error per 1 kb of amplification. Okay, so yeah, to avoid because you want to copy the exact sequence from the gene of interest and then to use it in your cloning, and maybe later on you want to express it, then you want an exact sequence. So you will need a high fidelity DNA polymerase. Also, DNA polymerase, some of them, they will generate blunt end, okay, which suitable for blunt end cloning. So when you choose for your DNA polymerase, also you need to double check, okay, whether this polymerase, it will generate a blunt end or a poly A tail, something like that. Okay, yeah. Okay, and for sticky end cloning, okay, choosing the restriction enzyme are very important. Commercially, you can find that there are conventional um, restriction enzyme, conventional RE. There are also upgraded version of RE. Yeah, usually we can have the high fidelity RE, something like that. Okay, so what is the um, advantage and disadvantage from the conventional RE and from the upgraded RE? Okay, a conventional RE, usually it will take 1 hour to complete the digestion, and different RE, they will require different reaction buffer. Okay, that which means if you are perform a multiple digestion that using 2 or more RE, you may need to optimize the condition of the buffer. Okay, or you may need to choose one buffer that is suitable for both RE, but with some give and take in the digestion efficiency. Okay, so possible that is lower digestion in efficiency in another RE, something like that. Okay, also for conventional RE, there are risks of star activity or when over digest. Okay, star activity, it means that the RE may digest at some unspecific site when the condition is not optimum. As mentioned just now, when you need to use another buffer or choose one buffer to perform the double digestion, then you may have a condition that is not optimum for one of the RE. Then it will have possible of star activity. Okay, yeah. Okay, so for example here, if you're going to use the AAT3 and ADE1 for your study, refer to this table. So you can find that the AT2, okay, AT2, it has 100% activity in this tangle buffer, while the ADE1, it have 100% activity in the green buffer. So it can also, the ADE1, it can also have 100% activity in tango buffer, but you can see that there is this star. So it means this tango buffer is not really a optimum condition for this enzyme. So it may have possible to have the star activity under this buffer condition. Okay, so if I'm going to perform a double digestion using these two RE, it means I can use tango buffer, okay, but with the risk of star activity. Okay, so if I don't want to have this, I don't want to use this tango buffer, I can also choose for this the 2 times tango buffer. Okay, but 2 times tango buffer, you will have the problem of the activity lower activity efficiency for these two enzymes. So the enzyme activity will be 20 to 50% only for both under the 2 times tango buffer. Okay, yeah, something like this. So for conventional RE, they will have this problem to choose the suitable buffer for the enzyme activity when you perform multiple digestion. Okay, yeah. So to avoid all this, you can also use Thermo Scientific fast digest restriction enzyme. So they can help you to overcome all the problem that is created from conventional RE, because the fast digest RE, it can complete the digestion in just 5 to 15 minutes. So it's very fast. You just need to wait for 5 to 15 minutes. Then you can continue your next step. Okay, so also it is very simple because it's using one buffer system. So for single, double, and multiple digestion, you can perform the digestion by using only one reaction buffer, regardless the combination of the RE that you choose. And you can choose from a wide range of 176 RE that is available under this fast digest enzyme. Yeah. Okay. And also the RE buffer, the reaction buffer, it is 100% compatible with any downstream application. Okay. And now it also comes with green buffer, which it gives you the convenience to direct load of the reaction mix into the agarose to run in agarose gel electrophoresis. So it is your handling where you want to run gel after the digestion. Yeah. Okay, so just now is for sticky end cloning. Now we have the blunt end cloning. So what you need for blunt end cloning, what you need to prepare for your insert in blunt end cloning. The DNA fragment, you may blunt it by filling the 5 prime overhang, or you chew back the 3 prime overhang, okay, to blunt the DNA fragment. So for this purpose, you can use either T4 DNA polymerase or clonal fragment from Thermo Scientific. Okay, the T4 DNA polymerase, it can be used for both purpose, while clonal fragment, it only suitable for filling in the 5 prime overhang. Okay, so now you have your insert ready for cloning. You need to prepare your vector also. Okay, so for vector, first we need to prepare the prevent the vector from recircularization. So vector, they are open usually. So sometimes they will recirculate. Okay, so for this purpose, you need to dephosphorylate the vector DNA. So this phosphorylation is the process by which phosphate groups are removed from both 5' end of the vector by using the phosphatase. Okay, removal of the phosphate group from the DNA fragment can prevent ligation. So after your vector is dephosphorylate, it means it cannot ligate with any DNA fragment anymore. Okay, so to perform this, you can use the Thermo Scientific Fast AP, the more sensitive alkaline phosphatase. Okay, so the reaction with this enzyme, it will only take you 10 minutes at 37 degrees Celsius, and only one protocol. Okay, so you need to only use follow that specific protocol, then you can perform it for all types of DNA and okay. But okay, how this process, how this dephosphorylation happen? Okay, a little bit on the basic. The DNA, in DNA synthesis, the 5 prime phosphate in the nucleotide, it will react with the hydroxyl group in the 3' end of another nucleotide to form the phosphodiester bond. Okay, so now if you take away the phosphate, as what I mentioned just now, your vector cannot uh, form any phosphodiester bond with any other DNA fragment. So it means uh, um, the insert, you need to have this 5 prime phosphate to facilitate the ligation, or else if your insert don't have the 5 prime phosphate, then ligation cannot happen. Okay, yeah, so do take note on this. And also 2 points to remember. One is RE digested DNA or insert, they will have 5 prime end with phosphate group. Okay, but the DNA that generate through PCR, so if you perform a PCR to get your gene of interest, then usually primer, they do not have the 5 prime end with phosphate, except you request to add this 5 prime end with phosphate in your primer when you order the primer synthesis. Okay, so it means primer don't have the 5 prime end phosphate, so your PCR product also do not have. Okay, yeah, so with this, it means that sticky end cloning, you will need to dephosphorylate the vector so that there is no background found in your transformation. Okay, so you need to dephosphorylate the vector, then there is no background found, but the ligation will happen. Okay, while for blunt end cloning, if the PCR product is direct use for cloning, So remember, PCR product, it do not have 5' phosphate. And if you perform dephosphorylation to your vector to uh, um, avoid the vector from recirculate, then you will have no ligation happen. It means ligation will fail, cloning fail. Okay, but if you do not dephosphorylate your vector, then there is possible of background in your transformation. Then possible you will have some false positive um, colony, okay, something like that. Yeah. So how? If you want to remain dephosphorylate of your vector to avoid vector recirculate rise, then you also need to use the PCR product for your cloning. It means you need to make sure your PCR product have the 5' phosphate group. Okay, so means you need to add the 5' phosphate to your PCR product. Yeah. So it means more work for your blind end cloning. Yeah. So under Thermo Scientific, there is a fast DNA and repair kit, which it actually include all the enzymes that are needed for blind end cloning of the insert and also dephosphorylate the end of the vector. So the kit, it includes the T4 polynucleotide kinase, the T4 DNA polymerase and crenal fragment, which can give you a fast and convenient end modification to your vector, and it is efficient that it can blunting and phosphorylate the DNA that has a low amount. Okay, yeah. Okay, so for after the insert and vector are prepared, you need to make sure they are clean and pure before perform your ligation, and to make sure there is no inhibitor that will inhibit the ligation activity. So you can perform your insert analysis or quantification after the RE digestion, and if you find there is like your PCR product have multiple band, then you can consider to use, sorry, Thermo Scientific CloneJet gel extraction kit to perform the purification. Okay, yeah. So now you get your pure insert and vector that is ready for ligation. So ligation, I can say is the most simple step in cloning. So it is a process of phosphodiester bond formation between the, as I mentioned just now, the 3' OH and the 5' phosphate group of the vector and insert. Okay, so this reaction, it can be performed by using the Thermo Scientific T4 DNA ligase. And some tips to share with you is to optimize the ligation reaction, you can try different molar ratio of the insert to vector. So commonly people will use 3:1 ratio, which means it refers to 3 portion of the insert with 1 portion of your vector in the ligation reaction. Okay, so this is the most commonly used. But if you still find maybe your ligation is not really working very well, so maybe you can adjust the ratio to put in more insert like 4:1, 5:1. Okay, so it can be up to 15:1, something like that. Yeah. Okay, so you can also increase the time of the ligation at the 37 degrees Celsius, or perform the ligation overnight at the 16 degrees Celsius to enhance the ligation process. Okay, yeah. Okay, so Thermo Scientific rapid ligation kit, it can also help you in your ligation. It can give you a fast ligation process that is complete in 5 minutes. Okay, so compare with conventional ligation, which usually it may need 15 minutes or up to 1 hour, something like that. Okay, so with this kit, the rapid ligation kit, you can complete your ligation within 5 minutes only. Yeah, 5 minutes. Remember this, 5 minutes only. Okay, and it is suitable for sticky end and blunt end cloning. And very important, the reaction mix, it will not compromise the transformation process that you are going to perform later. So this is very convenient to use. Okay, yeah. So after ligation, you assume that the insert has ligate to the vector, and we have get the recombinant plasmid. So we need to transform it into a host cell to get a more replicate of this plasmid. Okay, yeah. Sorry. Okay, transformation, it actually refer to the uptake of the recombinant plasmid into a host cell. There are many types of host cell can be used. So bacterial cell, it is the most commonly used because it can give fastest and efficient growth under also in a lower cost, and the culture process for the bacteria is very simple. Okay, but bacterial cell, it may not suitable for study on expression in some mammalian protein of the some of the mammalian protein. So for this type of study, a mammalian cell may be more suitable for the host as a host cell for the transformation. Okay, yeah. So the rest of the cell, they are less common to use, I can say, but some highlight is yeast cell. It is suitable for eukaryotic protein processing, but it's not easy to optimize the growth condition. Okay, and if you're working with insect cell for transformation, it may consume your time in optimizing the culture condition, and also for mammalian cell also. So the culturing of this cell, yeast cell, insect, and mammalian cell is um, more time consuming, and you need to perform more optimization. Yeah. So we go to bacterial cell um, transformation. Okay, because bacterial cell is the most commonly used host cell, and because it grow faster. So there are two methods that is used in bacterial cell transformation. One is using heat shock method, and another one is using electroporation method. In heat shock method, the bacterial cell, they will treated with calcium chloride to facilitate the attachment of the plasmid to the cell membrane. Then it will perform the heat shock to produce a pore in the cell membrane. Then it forces the plasmid to go inside the cell. So this method is much more easy, but it only need to use the water bath where you need to have the perform the heat shock at 42 degrees Celsius. So you need a water bath. Okay, but the transformation efficiency for this method is lower, and more DNA will be required for this method also. So it takes time to produce the competent cell for this heat shock method. In the meanwhile, for electroporation method, um, it used the electrical shock to create holes at the cell membrane, so that also it can force the plasmid to go into the the vector, the whole cell. Okay. But the problem is for this method, you will need a special equipment which called the electroporator. So electroporator, it will help to generate the electrical shock to the cell. Okay, so but this method is suitable for all types of bacterial cell, which include the gram-positive bacterial cell that have cell wall. And the transformation efficiency is higher also with this method. Okay, so one problem is you need the electroporator. Okay, yeah. Okay, so to ease the preparation of the competent cell, we have Thermo Scientific Transform 8 bacterial transformation kit. So it can help you to overcome all the hassle in handling and give you a high transformation efficiency. So this kit is very convenient where you can actually use the cell from overnight culture or from bacterial colony as the competent cell. Okay, so it's fast and easy to use. And under Thermo Scientific, we also have some competent cell. Okay, different type of competent cell. So it depends on your application. For example, if you want to perform um, cloning for large plasmid, so you can go for DH10B competent cell. Okay, so this competent cell is also suitable for bacterial artificial chromosome backmid or site-directed mutagenesis study. Okay, so if you're working on protein expression, okay, you may want to consider for this BL21DE3 competent cell. Okay, yeah. Also, we have a cloning kit that come with competent cell and also site-directed mutagenesis kit. The fusion site-directed mutagenesis kit that come with competent cell. Okay, yeah. So we have these two kit that come with competent cell also. Okay, a little bit on the tips for bacterial transformation. So first, for the competent cell, you can actually prepare your competent cell a few days earlier, okay, but avoid it to have many cycle of freeze thaw, okay, because freeze thaw, too many cycle of freeze thaw, it will lower the transformation efficiency because the competent cell is actually quite weak. The cell is actually quite weak because that is already pore in the cell membrane. So if you freeze thaw it many times, then the cell will die. Okay, yeah. Okay, also to improve the cell viability, the clone and also the cloning efficiency, you can culture the transformed cell in a antibiotic-free liquid medium for a short recovery period, like half an hour, which means after the transformation, you have the cell uh, the competent cell that is already have performed the transformation, you culture it in something like 500 microliter of the culture, okay, for a short period. Then you can put this 500 microliter all into the culture bottle. Then you culture it again. So the culture 500 microliter of the culture, you can make it antibiotic free. So for the cell to recover, and then only you culture it in uh, culture medium that have antibiotic. Okay, yeah. Okay, so far from all that I shared just now is more on perform the cloning manually. So you find that there is many things that you need to consider, you need to optimize throughout the whole process. And to simplify all this process, you can consider to get a cloning kit which have everything inside. Okay, so with cloning kit, it's more easier. You just need to follow the protocol to do the cloning. Yeah, so here we have the Thermo Scientific CloneJet cloning kit. Okay, a little bit on the traditional selection with blue white screening. Okay, um, traditionally, conventionally, many of us will be using a blue white screening method. So, under this screening method, the vector used will carry a LAGZ gene. Okay, so you can see here the LAGZ gene. Okay, so it will code for beta galactosidase. Okay, so this LAGZ gene, it will code for beta galactosidase, which it will hydrolyze X scale to a blue pigment. It means in your plate you need to have the antibiotic X scale and IPTG. So these two, the X scale, the LAGZ gene, it will hydrolyze X scale into a blue pigment. Okay, yeah. So when you perform cloning, the insert, it will insert into this LAGZ gene. Okay, so it means that when there is insert, this LAGZ gene, it will be break into two parts. Okay, so it will not express. So the um, after the transformation, you will find that the colony that carry insert, it will be in white color. The colony that do not carry insert, it means it will have this beta galactosidase express. So you will have complete beta galactosidase from this exact gene. So it will hydrolyze the X scale into blue pigment. So any of the blue colony, they do not have the insert. Okay, yeah. So one problem from the blue-white screening example, let's look at this picture. You will think that all this, the blue color, all this is the blue color, right? So this is possible to be a white colony. This is possible to be a blue colony. So you will see blue and white colony inside this half plate. And then when I let you see another side of the half uh, plate, actually this half plate is the one that is white colony, is the one have insert. This part is actually is a blue colony which they do not have insert. So you can see sometimes the blue color, it can also looks like a white color colony. So it will be quite difficult to differentiate. And this is the problem for blue-white screening. So now under Thermo Scientific CloneJet cloning kit, there will be no such problem of selection, blue-white selection. Okay, this kit is quite user-friendly, I can say. So it has all the component that is needed for cloning, including the vector, okay, the PJET 1.2 blunt vector, and the cloning, it can be performed within 5 to 10 minutes with at least 99% of the positive clone determined. So it is suitable for both blunt end and sticky end cloning. Okay, so there is a lot of restriction enzyme site that you can choose for your sticky end cloning, or you want it to be a blunt end cloning also can. Okay, yeah. So the vector PJET 1.2 blunt is a is specially construct with the multiple cloning site. Okay, in the middle of this this legal restriction enzyme gene. Okay, so there is a ECO 471R gene. So this gene is a leader restriction enzyme gene. So when there is no insert here, this gene will be expressed, and then it will kill the host cell actually. Okay, yeah. So in the other hand, if there is insert in this gene, so it will break this gene into two. So it means the host cell that carry this plasmid, it will survive. Okay, yeah. So it's much more easier for the selection. So that's why, as I mentioned just now, by using this kit, you will have 99% positive clone. Okay, yeah. And here also, I want to share a special extraction kit from Thermo Scientific, the Thermo Scientific Alligator cloning kit. Okay, so it's specially designed for protein expression study when you need a tight control of gene expression on the gene which you clone. Uh, like example, in some study, the gene of interest, they may produce protein that will toxic to the cell. So if expressed in high level, so you need to control the expression of this gene so that it won't be present in too high, expressed in too much in the cell and then it kill the cell. Okay, so in that case, it is suitable to use this kit. Okay, as the name of the kit, alligator, it is using a LIC, okay, ligation independent cloning system. So the PCR product uh, will insert into a target vector without without using any enzyme or ligase. So it works like by just annealing the long complementary overhang at the insert and to the vector. Okay, so with the LIC system, it will give you efficient cloning with low background. Okay, so and the handling is very much easier, which it involve only PCR and cloning. Okay, so no restriction enzyme, no RE will be involved in this by using this cloning kit. So how this how this LIC system work is, first, you need to create the PCR product with a complementary overhang. Okay, this overhang. Okay, so first you need to design your primer with to carry this overhang. So you need to refer to the kit instruction first. Okay, then use the two activity that is imposed by T4 DNA polymerase, the exonuclease activity, which in the absence of the NTP, it will chew the DNA from 3' to 5'. And the polymerase activity, with the presence of the NTP, it will add the nucleotide from 5' to 3'. Okay, so when you treat the PCR product with T4 DNA polymerase with absence of other DNTP, but there is DGTP only, then the polymerase, it will chew the sequence here up to this point. So when it found this point, because there is DGTP present, so it will have the polymerase activity and to add in this G. So with this, it will stop the digestion, the chewing of the sequence here, and you will have the overhang in your PCR product. Okay, yeah. So come together in this kit is the pellet vector. So this vector, it has the T7 promoter which control the level of gene expression. So the vector, it will carrying the EK gene and the and enterokinase gene or WQ gene protease recognition site to remove of his tag after the expression of the protein. Okay, so this plasmid, it must be transformed into an E. coli strain which have the T7 RNA polymerase gene, so under the control of an inducible promoter, so such as you can transform it into BL21DA3. Okay, yeah. Okay, so here to summarize the Thermo Scientific webinar that we have for so far. Okay, so Thermo Scientific, we have a bunch of products that help you in your molecular cloning workflow or your molecular study workflow. Okay, from DNA preparation to generation of recombinant plasmid and transformation. Okay, so um, even more simple, you can use the cloning kit that is available also. Okay, and meanwhile, to complete the whole process, also we have the fusion DNA polymerase that can give you a high fidelity PCR. We have DreamTech for your PCR, daily routine PCR. Okay, and also we have other um, gene ruler. Okay, we have agarose, we have TAE buffer. Yeah, so it have a complete set for your molecular studies. Okay, so do contact us for your if you plan for cloning or if you perform any molecular study. Okay, yeah. So here come the end of my sharing. Thank you.